is, what proteins? What about proteins? Want to repair their proteins, and how are the proteins damaged? They're denatured. Wouldn't we like to renature those proteins? Wouldn't we like to make them fold back into their proper status? It turns out that virtually every cell has built into it a mechanism for doing that. Okay? So cells get stimulated by heat to produce proteins that aren't otherwise being made. They're called heat shock proteins. And those heat shock proteins, when they're synthesized, help misfolded proteins to fold properly. Yes, sir? That's for the ones that don't automatically fold properly. That's correct. I'm sorry? I'm not sure I understand your question, Andy. No, prions don't misfold because of heat. No. So, but we could see it as an example of prions where misfolding could be a real problem. And so it would be in the cell's interest to keep folding good as much as it can. So these heat shock proteins play a role in that. Yes, sir? They are a type of chaperones. So the name for these proteins, I'll say it here since he brought it up, they're called chaperones. All right? Chaperone. Yes, sir?
What's the limit to these? Their maximum capacity they can do this?
Well, how would I measure it? So I'm sorry. His question is, what's the maximum capacity? There's not a way to measure that, essentially. Okay. So if the question is how high of a temperature, no. It's going to be that if the cell is able to mount a response, it will mount a response and try to do that. Otherwise, the cell will die. So it's a difficult problem to quantitate.
Can you measure the amount of the heat shock proteins that are made?
You can measure the amount of the heat shock proteins that are being made. Yes, you can do that fairly easily. Yeah. Yes, sir. Maya.
Are the chaperone proteins more stable and harder to denature then?
Are the heat shock proteins more stable and harder to denature? That's a very good question, Matt. You know, I don't know the answer to that question myself. What would you predict?
Probably if they're there still.
I would predict that as well. Yeah. The heat shock proteins do something cool. The chaperones, what they do is they make a little chamber. And that little chamber is where these proteins that need to be folded go into. And in that chamber, they're isolated from the rest of the world. It's kind of like timeout for proteins. Okay? You put them in there, they get a chance to think, and they fold properly, and they come back out, and it's true, and they come back out and they're folded properly. So, yeah.
Is there a temperature that when it gets back to its regular temperature, it needs to stay, or
So I'm giving you a little bit of a misleading picture. So let me clarify that for you. So you are making chaperones all the time. So you will either make more or you'll make other ones, and there's typically other ones that you make as you get this response. So if you have a heat shock, you'll start making a class of these that you weren't making before. Okay? And yes, after the cells had a chance to recover, those proteins will stop being made. Okay, good questions. All right, so, and I mentioned heavy metal the other day. Here's a heavy metal one right here. Remember I said that cells get exposed to heavy metals, and they need to be able to deal with them? There's one. And you don't need to memorize these. I'm just showing you these as examples. All right. That's more than we need here. Okay. Yes? I'm sorry? Well, when has the denaturing gone so far that it can't come back together? Okay. Remember that, of course, we have, it's not something I can give a number value to, but remember that we need enzymes to catalyze reactions. So if we denature any critical enzyme that the cell needs to be alive, we're dead. That's what it means. So if that cell has got a critical enzyme that it needs to be alive, let's say it's a transcription factor that needs to bind DNA to make something that we need, and that transcription factor is misfolded, and I can't make that stuff, I'm dead. And not I'm dead, but that cell's dead. Okay? So that's an example. There's many ways that a cell could die with that. Okay? If the cell had a protein that it needed to make ATP right now, and that protein's misfolded, the cell's dead. So there's a variety of ways that could happen. Yes?
Do heat shock proteins only get generated if the cell's at a certain temperature? Like, does it wait for the heat to pass?
Yeah. So I'm not sure I understand your question, but I think what you're saying is, does it only happen when the temperature is high? Is that what you're saying?
Like, does it, no, it's like if you're still burning, it's not going to start. Like, you know, like
Yeah.
the temperature
Yeah. So if you were at an elevated temperature where you were denaturing proteins, you couldn't stay at that temperature for very long. Okay? So I'll give you an example. One of the things we do in transforming bacteria is you can put DNA into bacteria. And the way you do it is you give them a little heat shock. And you give them a heat shock for about 2 minutes. And that heat shock goes from 37 degrees centigrade to 42 degrees centigrade. If you take them above 42 degrees centigrade for even a few seconds, they're dead. So that range is fairly narrow, and the time they can tolerate it is fairly minimal. So there's not a lot of flexibility. That's why I say it's a middle point. We go too much, those cells are dead. But somewhere in the middle, we can actually have cells that will be able to recover from that, which is kind of cool. But it's not a very wide range. I mean, if you're a human being and you've got a fever of 108, you're in bad shape. Okay? That's bad shape if you're at that point, because you're losing all kinds of stuff at that point. Okay. All righty. So your book, for reasons unknown to me, decided at this point it's okay to talk about modifying RNA. So since I do what the book tells me, I guess I will modify RNA as well. We're now talking about what happens to RNA after it's been made, not how we're making it. Yes, sir. Sir. No, I'm not at the end yet. This is, yeah, I'm sorry. So we'll go through this. This chapter is what I'm talking about. Okay, good question. Okay, modifying RNAs. All right. Turns out RNAs are significantly and chemically modified after they're made. The modification occurs typically more often in eukaryotes than in prokaryotes, but both get modified. Okay? One of these that gets modified are the transfer RNAs. Transfer RNAs get modified in both prokaryotes and in eukaryotes. And the modifications are odd. They're very odd. They involve putting various methyl groups on, various amine groups on, in some cases converting uracils to thymines. Okay? And you wonder why they didn't just put it in the first place. Some very odd modifications that happen. And we're not going to focus too much on what those modifications are. But I do want you to know that chemical modification of those does occur, and it occurs in both prokaryotes and eukaryotes. When we look at a transfer RNA, it has a sort of a cloverleaf structure, and you can see that cloverleaf structure right there. Transfer RNAs are made as a longer molecule that's cut out of a bigger piece of RNA. So they're made as a long RNA, and then pieces of them are chopped out. So it's a chopping process that's cutting it out of a bigger RNA. So here's a bigger RNA that gets chopped, and we get some things arising from that. Okay? The 3' end of the transfer RNA gets chemically changed to the sequence CCA. This is true in both prokaryotes and eukaryotes. The CCA at the 3' end of the transfer RNA is important. And it's on that CCA, you can see it down here, it's on that CCA where the amino acid will be attached. So the amino acid gets attached to the CCA at the 3' end of the transfer RNA. Okay? And that's about all I want to say with those. Messenger RNAs aren't really modified in prokaryotes. There's very, very little modification of messenger RNAs in prokaryotes. However, in eukaryotes, there's a lot. A lot of things happen to eukaryotic messenger RNAs. Okay? One of these things is at the 5' end of the messenger RNA. This only happens to messenger RNAs in eukaryotes. It doesn't happen to transfer RNAs. It doesn't happen to ribosomal RNAs. It only happens to messenger RNAs in eukaryotes. This modification changes the 5' end of the messenger RNA. It puts what's called a cap onto that 5' end. And that cap is a modified guanine nucleotide that has a very, very unusual link. It's a 5' to 5' linkage. The 5' end of the modified nucleotide is covalently linked to the 5' end of the messenger RNA. Now, the effect that this thing has is it actually serves, first of all, to flag, hey, I'm a messenger RNA. When the cell sees that cap, it knows it's got a messenger RNA. And messenger RNAs are translated. That's useful information. So having that cap there is an identity tag, partly. The ribosome, which comes along and is going to translate that, grabs a hold of that cap. In prokaryotes, there's no cap. They just grab and they start translating. Okay? No identity tags. The other thing that the cap does is it provides stability to the messenger RNA. When I first started talking about RNAs, I told you that RNAs aren't around for a long time. They get broken down. Cells don't want to have RNAs around forever because it might have a need for a protein for a while, but later it doesn't need that. It doesn't want to have that around. So the cell is full of nucleases that will break down RNAs. This provides a protection against nucleases. So the 5' end is protected. Okay. So the cap provides protection and identity. That's what it does. Now if we think of the other end of the messenger RNA, something interesting happens there as well. And this only happens in eukaryotes. So only in eukaryotes do we see this other modification to messenger RNAs. And this modification involves putting what's called a poly-A tail on the 3' end of the messenger RNA. A poly-A tail. Well, as its name would suggest, it's a series of many, many, many adenine residues attached to the 3' end of the messenger RNA. And like the ones at the 5' end, it provides identity. It says, hey, I'm a messenger RNA because this doesn't happen to other RNAs. And it also provides protection against nucleases. Now, it provides protection in a little different way. It's interesting. The longer the A's, and in a typical messenger RNA, you might see 200 to 300 A's put on the end. The longer the number of A's that are put on the end, the longer the messenger RNA can be detected in the cell. What does that mean? Well, what it means is that the degradation that happens of messenger RNA when it starts getting broken down by the cell starts at the 3' end. And it works a few bases away and the nuclease falls off. And it works a few bases and the nuclease falls off. The longer the number of A's, the longer it takes the nucleases to chop up the messenger RNA. So more A's provide more protection. Cells don't want to have complete protection because they do want to break down those RNAs at some point. So the message the cells have is the longer the sequence I put on there is, the more long I want to use it. And the fewer I put on here, the less time I want this thing to be around. Make sense? Clear as mud? Okay. I'm guessing it is. All right. Now, the last modification that I will mention here, I've talked about before, and I'm not going to go through the steps of it, but I have pointed out that RNAs get spliced. We talked about splicing. That is a modification. With splicing, we're taking out sequences called introns, and we're joining the pieces that are left into exons. Splicing only occurs in eukaryotes. Splicing only occurs in eukaryotes. Does not occur in prokaryotes. We're not going to go through the steps of splicing. Okay? So if I ask you what an exon is, I trust you could tell me. What's an exon? Besides who's out in the Gulf right now, right? So what's an exon? So it's the part of the messenger RNA, okay, that's remaining after the introns are removed. And as Matt said, it exits the nucleus and goes and gets translated.